My name is Sapir. I'm coming from the wonderful Meital Oren's lab. And I want to start my talk today with an assumption. And this assumption is that almost everyone in this room knows at least one person suffering from some type of brain disease, or what we call neurodegenerative diseases. So for example, we have Alzheimer's, Parkinson's, Huntington's. I myself have a young sister coping with multiple sclerosis. And the reason for this assumption is that these diseases are extremely common. In Israel alone, hundreds of thousands of people suffer from it. Now, a personal story, when I first entered the world of biology, people around me kept asking me if I found a cure for cancer yet. And I said, no, I'm not even working on cancer. And then they asked, and then 2020 came, and they asked me, have I found a cure for COVID yet? And I said, you have no idea what I'm doing in life. And then when I started in the world of neurobiology, people asked me if I found a cure for Parkinson's yet. And my answer is no, and it's not only my answer, it's the objective answer. There is no cure for these diseases. And there are two parts as to why. First of all, these diseases occur in the most complicated organ in our body, the brain. And not only that, but in order for such diseases to develop, different factors need to work together, such as the environment we live in, nutrition, what we eat, different toxins like alcohol and cigarettes, and genetics, or our genes. Now, one such gene that has been associated with different types of brain diseases and disorders is DCC. And when I say associated, I mean that mutations in this gene have been correlated to the possibility to have one of these diseases. So diseases like Parkinson, OCD, anorexia, schizophrenia, and more. DCC generally is most well renowned for its role. It's a receptor, and it's most renowned for its role in the organization of the developing nervous system during fetal development. So if you take, for example, more specifically, one of its important roles is in what we call axon guidance. It helps neurons reach their target. So if you look at this example here, we see that this neuron normally is supposed to go ventrally or down and then anteriorly or to the left. But when DCC is mutated, there are defects in this guidance, and we see that it does not go ventrally and only anteriorly. Okay, this is like what it is most renowned for. Because of its role in fetal development, it's highly expressed during fetal stages, but the expression diminishes as we grow older. However, in some parts of our brain, the expression of DCC remains. And if more specifically we look at the midbrain, which is a certain region of the brain, we see that it remains in high expression there during adulthood. And we don't really know what is its role during adulthood. Another interesting and important fact I want to tell you about the expression of DCC is that not only is it highly expressed in the adult midbrain, but it is expressed in cells that produce dopamine. So specifically in dopaminergic cells in the midbrain. And the reason this is interesting to us is because these are the exact cells that undergo degeneration first during the early onset of Parkinson's disease. Okay, so these dopaminergic cells that express DCC are the first to die during Parkinson's disease. Now, if we see a person hanging around in a crime scene, we immediately assume he has something to do with the crime, right? And we would like to investigate him. And this is exactly what we are saying. We are assuming that DCC is associated with Parkinson's disease because of its location, and my question is how? Now, in order to answer this complicated question, we undertake a holistic approach in which we use various models. We use human dopamine producing cells in cell cultures. We use mouse model, and we use C. elegans, which is what I'm going to be talking about today. So C. elegans, small, round, transparent worm. And C. elegans offers us a very powerful way to simplify the tool in order to answer very complicated questions. It's very complicated to study the brain, even just the midbrain. And if we downscale it to a relatively simple organism that we can genetically manipulate relatively easily as well, we can get a molecular insight but still stay in the level of a whole organism. And this is what I want to show you today. So again, this is our lovely C. elegans. And if we focus on its head, we can see that it has dopamine-producing cells. This is what we see here in green. So this is the cell body, and these are the dendrites that it sends to the tip of the nose of the worm. So these are the dopamine-producing cells, and we have ways to mimic Parkinson's symptoms and in our case, the specific death of dopaminergic neurons. Okay, and so I've used one of these models. It's a toxin that when we induce it, when we introduce it to the animal, we see a death specifically of dopaminergic neurons, and this is what a sick neuron looks like. Okay, we see loss of the dendrites, we see these blebbings, what we call blebbings. Okay, now I told you that mutations in DCC has been associated with different diseases, and so our hypothesis was that by mutating DCC to a loss of function mutation, making it lose its function, we actually would see a worse phenotype of the disease. We see cells perhaps dying faster or more severely sick, and much to our surprise, the exact opposite happened. When we took animals with a mutated DCC and introduced them to the toxin, we actually saw a protective effect. These cells were much less sick than the control ones. Now, the other way around, we have a way to manipulate the expression of DCC so it will remain highly expressed. And again, to our amazement, not only were the animals sicker, but in fact, even in the absence of the toxin, just by stabilizing the receptor, we see death of specifically dopaminergic neurons. And this was very cool to us. And we try to understand why or how are the cells dying. We know, or I'm telling you now, that there are different pathways of cell death. And there is one type of alternative cell death called partanatos, which has been very recently, it's very novel, been implicated in Parkinson's disease. And we were wondering perhaps partanatos is what takes place here when we stabilize the DCC expression. So again, this is the stabilized DCC. We see the sick neurons. And in order for us to understand whether partanatos is involved, what we can do is we can inhibit factors in this pathway and see if we can inhibit the death of the cell. And you will believe me when I tell you that PARP1 and WA1 are parts of factors of partanatos. And indeed, when we inhibit or silence, cells are not dying anymore. And we are reversing the phenotype, meaning the stabilization of ANC40, of DCC, sorry. ANC40 is the worm homolog. The stabilization of DCC causes a dopaminergic cell death via partanatos. The last thing I want to tell you about, we know that these brain diseases usually differ between men and women, differences in terms of prevalence, severity, progression of the disease. And we were wondering whether we can recapitulate this in such a simple organism such as C. elegans. And so everything I've shown you today was in a female worm. And again, this is a female worm with stabilized DCC, and the neurons are sick, and this worm doesn't cease to amaze us because males are not affected at all. So in such a simple organism, we can recapitulate what we see in such complex organisms such as mammals. And so allow me to sum up what I've shown you today. I've shown you that a loss of function of DCC, the DCC receptor, leads to a protective effect against the Parkinson's disease model. I've shown you that just by stabilizing DCC, we get Parkinson-like symptoms, meaning the specific death of dopaminergic neurons. I've shown you that this occurs via an alternative cell death pathway, partanatos, which has been recently implicated in Parkinson, and that this effect differs between males and females. I want to very much thank my amazing lab, which is like a family to me, and I want to thank you for listening. And I'll be okay with taking questions.
You did it.
Without
Thank you very much for the great talk.
Thank you.
Great question. Yes.
Do you have any explanation to the counterintuitive phenotype that you see?
So it's counterintuitive because I said we see mutations in mammals or in humans, but we don't know what the mutations are. What if the mutations are stabilizing? So that's one explanation. That's perhaps what's going on, and that's what we're trying to find out.
How are you investigating DCC in the mouse model?
So the way to stabilize DCC in the worm is Yuda Zalzberg in our lab has done the work, is to manipulate a conserved regulatory site. And this is what we're trying to do in mice now, or we are doing via CRISPR. And we are trying to stabilize the receptor and then try to understand what happens in mammals when we do so. And hopefully, this will lead to similar phenotypes like the specific death of dopaminergic neurons. This would be like groundbreaking. Yeah, to me.
How common is that you see really the negative effect for male and females, and is it how it affects like how we give drugs, right? For you need to be like also relates to personal medicine and everything. But how common is this in neurodegenerative disorders?
The difference between men and extremely common. For example, in Alzheimer's, it's much more. Or let's talk about multiple sclerosis. My sister. It's 4-1 in females. The age of onset is earlier in females. It's in women. It's like in almost all neurodegenerative diseases, you would see a difference between men and women.
What is the explanation?
That's what we're working on. Yes.
Lately, it's been said that long COVID has to do or show some kinds of symptoms like in neurodegenerative diseases. Do you have anything that relates in your research to what is being said right now?
Unfortunately, not in COVID, no. Sorry.
Long COVID.
Not in long COVID. Yeah?
Yeah, thank you for an excellent talk.
Thank you.
Have you found some receptor beside the DCC that can do the same things or just unique?
I mean, no, we haven't looked. That's the truth. We got to DCC via a prior project in our lab, and then I stepped in and we tried to understand its relations with dopaminergic neurons, and then we found out this amazing. But we haven't looked at other receptors with similar.
The phenomenon in male is exactly the opposite when you do the loss of function, that it shows the opposite effect, or just the stabilizing is not effective?
So we haven't looked at the male reaction to the loss of function, but if I had to guess, I think it wouldn't have the opposite effect. This is what I think.
So you mentioned that you detected DCC expressions in the midbrain.
Yes, not me.
Yeah, but how do you do something like that?
Immunohistochemistry, immunostaining, again, like antibodies against DCC in sections.
Yeah, but so do you do it on live people, like in the?
No, you do it on mice and then on brain samples from brains donated to science. You cannot do it live. Okay, thank you.